Hi everyone, my name is Daniil Gataulin. I'm one of senior research analysts covering biotech here at Chardon, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Anirvan Ghosh, the CEO of Unity Biotechnology, to a virtual ophthalmology summit where the initial focus is on AMD and diabetes-related ocular diseases. Anirvan, thank you for joining. Welcome to our session.
Thank you for having me.
Great. So to start things off, could you provide a brief overview of Unity Biotechnology and perhaps highlight some of the recent milestones?
Absolutely. So Unity is focused on developing therapies that are based on our understanding of the biology that underlies aging, that is relevant for both age-related diseases and other chronic conditions. In ophthalmology, our focus is on targeting DME and AMD, which are two kind of leading causes of blindness. And our therapeutic approach is to develop senolytic drugs. So a common feature of diseases is an accumulation of dysfunctional cells in the tissue called senescent cells. And over the last several years, we have developed a platform that allows us to target those cells that lead to disease progression. And by eliminating those cells, we can achieve better outcomes. So that's the primary focus of Unity.
Got it. So as you mentioned, you are targeting senescent cells in retinal indications. It's a unique approach compared to what's being developed over the past many years from other companies. So for those unfamiliar with the approach, can you help explain what senescent cells are and why do you believe that removing them is important?
Yeah, it might be helpful just to give you a bit of a background about the biology that underlies DME and AMD, and then I can put the senescence approach in context. So both diabetic macular edema and age-related macular degeneration are diseases that have a primary vascular component. So the reason you lose vision is triggered by a problem in the vasculature of the eye, or the vasculature of the retina, really. And in the course of disease, you end up getting leaky blood vessels, which can lead to fluid accumulation in the eye, eventually get to ischemic areas so that the underlying tissue is damaged and the disease progresses. The standard of care treatment in this field for many years, over a decade now, has been anti-VEGF antibody treatment. And they've been quite successful and helpful drugs in the field. And they fundamentally work by preventing growth of blood vessels. So the idea is by preventing growth of abnormal blood vessels, you could have benefit. And in fact, you do see benefit in many patients. It turns out that in about half the patients in DME, patients do not get to 20/40 vision or driving vision, even with about a year or more of treatment, right? So there's significant unmet need in the field. It also indicates that there must be other elements of disease beyond VEGF-dependent drivers that lead to disease persistence. And over time, most people continue to lose vision. So even if they have some gain, it tends to be somewhat transient. So beyond the first year, there's progression of disease because the underlying disease is still active. In our case, our interest was to see whether senescent cells might play a role in disease progression, particularly in the vascular component. And what we found was that senescent cells, in fact, do accumulate in the blood vessels during these diseases. And in preclinical models, we found that if we target them selectively with a BCL-XL inhibitor, we can reverse many of the hallmarks of disease pathophysiology, which should lead to improved outcomes. So that's the motivation of exploring senolytics in eye diseases.
Got it. So the molecule that you're developing, your lead asset is UBX1325. And as you mentioned, it's being developed for DME and wet AMD. So can you talk a little bit about that molecule and its mechanism of action?
The UBX1325 is a selective BCL-XL inhibitor, BCL-XL being a protein that is required for the survival of senescent cells, but it's not required for the survival of healthy cells. So because of this differential requirement for survival, if you introduce a BCL-XL inhibitor into the eye, then that selectively leads to elimination of senescent cells while leaving healthy cells intact. So that differential activity is really at the core of having a selective senolytic. In our case, BCL-XL inhibitor 1325 is a small molecule that is introduced into the eye intravitreally, which is the procedure that's used for anti-VEGF treatment as well, something that physicians are very used to and comfortable with. But there is an expectation that in contrast to anti-VEGFs, which require persistent treatment because you have to actively manage the growth of abnormal blood vessels, that with 1325, we expect that you should have long-lasting effects after a single injection, because that should lead to elimination of senescent cells, and then your blood vessels should be healthy for a long period of time. So that is how we are approaching the development of 1325. In fact, our early results are consistent with that expectation.
Okay, and how is it administered?
Intravitreally, it's an injection into the vitreous of the eye. It's an outpatient procedure. The procedure typically lasts less than 15 minutes in a doctor's office.
Got it. Understand. So you've recently presented data from phase 2 Behold DME study. But before we talk about the results, can you walk us through that study design and highlight some of the patient baseline characteristics?
So what is very important for us, and I think important for any new treatment entering the field, is that it needs to be differentiated compared to the standard of care, right? So if I'm going to ask a patient to go on to new treatment, the benefit or potential benefit has to be clear. And in our case, therefore, our focus has been to clearly identify those patients who are not responding adequately to anti-VEGFs, and then see whether we can provide benefit to them. So the first element of that design was the selection of patients. I mentioned that almost half the patients in DME don't get to 20/40 vision. So we recruited patients who had been on anti-VEGF for at least 6 months and still had visual acuity deficits. And as a measure of active disease, we selected patients who had significant fluid in the eye. So when the drug is working well, when anti-VEGFs are working well, you get nice drying of the retina. Whereas if you have persistent fluid, it says that your anti-VEGF is not working optimally. So we selected patients who had fluid in the eye and who had visual acuity deficits. Just in terms of what those criteria were, to give you a sense, they needed to have fluid of over 325 microns in the eye. Our patients on average came in with over 400 microns of fluid, so quite a bit of fluid. And they needed to have a visual acuity of less than 73 ETDRS letters, a scale that's used for visual acuity. Commonly, in our case, their average ETDRS coming to the study was 60 letters. So patients coming in with really poor vision and fluid in the eye, although they had been on anti-VEGF for quite some time. We then took them off of their anti-VEGF and we randomized them to an active arm, which is the UBX 1325 arm, gave them a single injection, or to a sham arm, where they got a sham procedure that was indistinguishable in every way, except they didn't get an active drug.
Got it. And you completed the readout at 48 weeks. Can you tell us what you found?
Yeah, so the treatment protocol was to give them a single injection of UBX and then follow them out. The primary readout was actually at 24, and then we had a long-term extension to 48. So I can speak to both of those data readouts are quite consistent. And we also assessed, along with visual acuity, fluid in the eye, and the need for rescue treatment. This last point is important because in many of the newer treatments that are under development, there's a lot of interest in understanding durability. And the durability is typically defined in how long patients can go without needing a rescue injection, right? So those are the elements. So the most striking thing about this study was that in this patient population that had essentially plateaued on their anti-VEGF treatment, we found that patients who are on UBX 1325 had a gain of over 6 letters of vision out at 24 weeks, and that vision gain was also seen at 48 weeks. So the two primary points of analysis, 24 and 48, patients had gained significant number of letters. It's important to note that in this period, patients who have been on anti-VEGF longer than 6 months, if they remain on an anti-VEGF, they stay within about a letter of vision. So it's a very, very tightly controlled visual acuity scale. So instead of being right at that baseline vision, the patients on UBX had a gain of about over 6 letters compared to the sham patients who had no gain in vision at those time points. So that was the primary vision readout, which I think is quite important. Correspondingly, we found that in the sham patients, there was a gain in fluid during that period if they were not rescued, whereas the patients treated with UBX1325 were able to maintain their central subfield thickness and actually even showed a trend towards improving as you went towards the end of the study, which is pretty remarkable that these patients who went 1 year without anti-VEGF treatment were able to have visual acuity gains and no gain in fluid.
Got it. So one thing that you mentioned about anti-VEGF rescue criteria, and I think that's becoming somewhat an important question, especially in the earlier trials, is the consistency of those criteria. So can you tell us what those criteria were for your study, and how consistent were they among the physicians?
Certainly. So let me just start briefly with reminding people here about how the standard treatment is delivered. So they typically get 5 loading doses of anti-VEGF every month, and then they move to every other month. So that is the standard dosing protocol for aflibercept, right? So they would be on every other month dosing. With regard to rescue criteria that is common in the field, patients are generally rescued if they have a 10-letter drop from peak vision or a gain of 75 microns of fluid. In addition, physician discretion is always an option. So a physician who's following the patient can decide at a given time based on their other elements of disease activity that they feel the patient needs a rescue. So that's the framework. Importantly, in our study, as well as many prior studies, the criteria are an OR criteria. So the patients can be rescued if they have a loss of vision or a gain of fluid. For example, that is also the criteria that is used in faricimab, which is the latest approval in this field, which aims to get about half the patients to every 4-month dosing, roughly in that ballpark, and has been seen as an important entry into the market. I call that out because in a recent study of high-dose aflibercept, they shared their data in terms of an AND criteria. So the patients were rescued if they had a gain in fluid and loss of vision. And with that AND criteria, they showed a significant number of patients going out 6 months or longer. But that is not that commonly used. And we and analyzed our data as well to say that if you use an AND criteria, what would happen? And virtually nobody would get rescued over the course of 48 weeks if you use that criteria. Having said that, given the criteria I stated, what was also striking to us is over half the patients, over 50% of the patients went a whole year, went 48 weeks without needing a rescue treatment. So that was the durability benefit that we saw of UBX1325.
Okay, got it. And with respect to durability, it seems quite significant. What is it about the molecule or the mechanism of action here that you think allows for that prolonged effect?
Yeah, so we get this question quite a bit because people are used to treatments where the patients are being treated every other month or so and then having some benefit, in which in our case, we have a single injection, then we have this prolonged benefit. And we know that the drug doesn't stay in the eye that long. So we're not playing with the formulation of the drug, for example, that we just have it resident in the eye longer. So in our case, the prolonged duration is really best explained by a pharmacodynamic or PD effect of the drug, where the effect on potential disease modification is seen as prolonged activity. So the drug is in the eye for about 2 days, which is also actually important from a safety element, and hopefully we have a chance to discuss safety later in our conversation today. But the drug is in the eye only for a couple of days after that injection, and we expect during that time it will clear the senescent cells in the blood vessels. Then the drug is gone. Hopefully most of the senescent cells are cleared, and then you then see the consequence of that in terms of reduced leak, so you don't get additional fluid accumulation. And because of the improved perfusion through the blood vessels, you have improved vision, right? That's the way we envision the drug acting, which would explain how we have significant number of patients going long periods without needing additional treatment.
Got it. Got it. And in terms of data from that trial, if I recall correctly, there were some fluctuations in BCVA measurements, actually in both the 1325 and sham arms. Just wanted to hear your thoughts on what's causing those.
Yeah, so the BCVA measure is quite similar to a regular eye chart measure that you have at the optometrist office if you go for a vision exam. And in any person, normal person, or a person who has DME or AMD, if you assess them multiple times separated by a few weeks, often their performance will fluctuate a little bit. It's not uncommon. This would be true for us as well. This is true for all the patients who are on anti-VEGF previously. So any individual patient will often have some fluctuation. However, the fluctuation will move around a mean that reflects their true vision. Hence, it is important for us to really look at overall disease trajectory so that we look at the performance at multiple time points. I mentioned that 24 and 48 weeks were the primary predefined time points where we looked, but we did have assessments at 8, 12, 8, 18, and also between 24 and 48 weeks. So we can look at how the vision is doing at each of those time points. And what we find is what I described were kind of the mean improvements in vision are seen at multiple time points. At most of the individual time points, you see gain in BCVA in the UBX arm compared to sham. There are a couple of points where you see that the gain is not as pronounced in particular time points, and that is consistent with kind of historically reported variability in vision assessments in between visits. So that itself is not unusual, and we do in fact see it in the both UBX arm and the sham arm. I'll make a comment around the impact of rescues on the sham arm because that is important to note. So I mentioned that in the UBX arm, over half the patients went without any rescue, whereas in the sham arm, about 80% of the patients needed a rescue at some point. And right from the beginning, you can see that if a UBX patient needed a rescue, it was later than a corresponding sham patient. But the sham patients continued to accumulate rescues. And the result of that is that in the end, there are few sham patients who do go without needing rescue at the end, but those are really the best of the sham patients, if you will. They've somehow gotten to some stable disease. But the fact that 80% of them do need rescue indicates that this patient population is in need of treatment and is in a fairly active disease state.
Yeah, that makes sense. And in terms of safety, yes, I wanted to spend some time to talk about that. There were no intraocular inflammation events, I understand. But wanted to ask if there were any other common effects or adverse events that emerged with treatment.
Yeah, overall, we were very pleased with the safety profile of this molecule, and something that tends to be really important in the field. You'll note if you follow other ophthalmology studies that there is a huge amount of attention paid, particularly to intraocular inflammation, because it's a situation that can be hard to manage, and physicians really want drugs that ideally don't have IOIs. So going 48 weeks in this study and across all the studies we've run so far without any instance of intraocular inflammation is a really promising sign. So we're pleased about that. The adverse events in general that were reported were well balanced across the arms, and those that were reported were really mainly related to injection sites. So some, in response to an injection into the eye, you can have a transient something called a conj hemorrhage. That is a small blood vessel that can get damaged, but then that typically resolves within a few days. It can happen in the course of normal life if you happen to get hit or some injury to the eye, but does not require attention. So there's nothing that required any intervention, and they're all self-resolving and generally well balanced. So we're quite pleased with the safety profile of the compound.
Mm-hmm. Got it. And to summarize, what would you say were the key takeaways from that study that informed the design for the phase 2b Aspire study that I believe you're planning on initiating fairly soon?
Yeah, so for us, the highlights of the data set were that we saw evidence of a significant efficacy in patients who had already plateaued on their anti-VEGF treatment, right? So patients who had been on anti-VEGF for a while, still had a lot of fluid in the eye, still visual acuity deficits, and we were able to give them significant improvement in visual acuity and manage their fluid. So at a top line, that is consistent with the differentiated profile for this molecule. If that continues to be the case in future studies, I think that would be of high interest, and all the feedback from the KOLs and patients group has been very positive. They're excited about having a new mechanism of action that works in a different way, that could allow patients to have benefit beyond whatever they might be achieving with anti-VEGFs. We expect that anti-VEGFs will remain the standard of care for some time, but as illustrated in the study, there's a lot of unmet need even with anti-VEGF, and we think that this drug has a profile that could, fill that need. Going into the Aspire study, I said the main thing that we want to fully understand is the differentiated benefit compared to aflibercept, which is the standard of care. So in the previous study, we had run it as a sham control study for a couple of reasons. One is that it was the first time we were in an efficacy study in DME with this new mechanism of action. And we wanted to have a clear understanding of what the signal or the benefit of UBX1325 is in a sham control study before introducing aflibercept. For example, we did not know, would we have to plan on multiple doses? Could that potentially provide benefit? And anything about any run-in treatment that might be important in an aflibercept treated study, et cetera. So going into the Aspire study, the main thing we're looking at is to provide strong evidence of differentiated benefit compared to aflibercept, which is a standard of care. So the elements of the study, just to highlight, at the key is to have matched injections over the first 6 months. It's a 6-month study right now, 24-week study of UBX1325 versus aflibercept, and see how our patients do compared to that treatment. And the second feature is to have 3 run-in aflibercept injections before they're randomized to either UBX or aflibercept. So let me take those design elements in turn. So the first, as I mentioned, that the data set we just announced had a single UBX injection, and then we had significant benefit. So why do we have additional injections here? So there are 2 reasons. One is we wanted to make sure that we were seeing the maximum benefit of UBX in patients in this next study. So while we saw strong gains with a single injection, it is possible that a single injection does not clear all senescent cells, and that an added treatment could further add to letter gains, which would be, important for physicians and patients, and important for us to know. The second is that the general guidance from FDA over the past year about aflibercept controlled studies has been a strong recommendation of having the treatment matched during the initial efficacy period, so that one has a full sense of the efficacy of the test drug compared to aflibercept. So having them matched over those first 24 months allows us to show that, to say that if you have matched treatments, you can see this is what you'd get with aflibercept, and this is what you get with UBX1325. I should mention that this does not preclude the possibility of adding a long-term extension to the study, where we could let patients then go without needing additional treatment and establish durability. But the first phase will really be a head-to-head efficacy study. The other element is about the run-in dose of having these aflibercept run-ins before you randomize. And that is to make sure that we have really gotten patients to an aflibercept plateau, because patients coming in can be on a variety of anti-VEGFs. And we don't want them, switching from, say, Lucentis to aflibercept or something like that, and then having some unexpected vision gain that confounds our analysis of our data set. So we'll have these run-in injections, and then after that, we'll take them off with that run-in, randomized to UBX or aflibercept.
Got it. And do you still expect to initiate the study in the second half of this year?
Yes, we're on track to initiate in the second half of the year. I'll mention many of the sites are the same ones that we had in Behold, and all those physicians were very excited about being in the next study, which is good for us in terms of, being able to stay with our recruitment timelines.
Understand. Excellent. And in terms of the efficacy, are you at all concerned that 24-week period may not be sufficient for senolytic mechanism to fully exert itself?
No, we are quite confident that that should be a long enough duration for us to see the benefit of UBX. In the Behold study, we started seeing the treatment effects separating from sham between 8 and 12 weeks. And by the time we got to 16 weeks, the signal was quite strong. At 24 weeks, it had gone to 6 letters, which is also then maintained or seen at 48, right? So we saw that even with a single injection, we got to maximum treatment benefit by 24 weeks. In this case, we'll have a couple of additional injections. So we'll see whether that leads to additional improvement, but we expect that at 24 weeks, we should have a good sense of the treatment benefit. I should also say historically, by 24 weeks, even with anti-VEGF treatments, one has a pretty good sense of the benefit that drug would provide.
Got it. That makes sense. And so at week 0, do I understand correctly that patients receive both 1325 and either aflibercept or sham injection?
Yeah, so this is an additional detail on the UBX arm, which is that at the very first visit to UBX, they get an injection of aflibercept as well as UBX. And the reason we did this is that in the Behold study, we had noticed that there were some rescues that accumulated in those first 8 weeks, and that the UBX benefit really started showing up between 8 and 12 weeks. So there is a period when if you remove patients from anti-VEGF, then there is a possibility that some patients will need rescue early on as a result of anti-VEGF withdrawal. And we wanted to minimize that. And so by giving them an injection of anti-VEGF in the first injection, it allows us essentially to traverse that period of the first 8 weeks until the UBX effect kicks in. And we expect the effect of that should be to reduce the rescue rate further in the UBX arm. So that's the rationale for that particular first injection.
Got it. And from the safety perspective, are those done on the same day? The concern is, the injection volumes are somewhat high. Are there potential for increased intraocular pressure, for instance?
Yeah, so the dual injections happen in the same visit. The patients get an injection of anti-VEGF, then they wait for about 30 minutes, and the physician watches for any change in IOP, and if things look good, then they can get the 1325 injection. I should say that we've had quite a bit of experience with that dual injection procedure in our Envision study, which is a study we ran in AMD patients, and there were no issues. So every patient slated for the 2 injections was able to get the injections. And so the physicians and patients were quite comfortable with it. It all happens in one physician visit, so we don't expect there to be any problems. And in this case, we're only doing it at the very first visit, and after that, they just have single injections.
Got it. Okay, that makes sense. And switching to AMD, do you expect, given the mechanism, that senescent cells would play as large a role in wet AMD as it would in DME?
Yeah, so in AMD, we ran a study called Envision that was designed as a head-to-head study against aflibercept, and has 2 parts to it. The part A was just a direct comparison against aflibercept, and part B has a combination arm to see whether there's added benefit if you combine the 2 drugs. Based on our prior data, we also see a rationale in AMD. There's senescence burden in AMD. And one of the things that we know from the studies which we previously shared is that senescence burden increases over time in AMD patients. So in the first couple of years, the burden is low, but then beyond 2 years, the senescence burden continues to increase. In the study that we shared on AMD, we took patients, any patient that had had AMD for more than 6 months were eligible and came into the study. And what we noticed that in the overall study population, we actually did not meet our non-inferiority criteria against aflibercept. And the analysis of data provided some insight in terms of what might have happened, because in the UBX arm, we actually maintained vision of those patients quite well. So they went 24 weeks without needing significant rescue and maintained vision. But on the aflibercept arm, there was an unexpected gain of about 3 letters after the first study dose. And so now what we see is that that seems to have been because of inadequate run-in. So they had one run-in injection of aflibercept, then came into the study. If they had had 2 injections, there would have been no additional letter gain. That actually informed our design of having 3 run-ins in the Aspire study so that we don't take any risk in having any letter gains in the aflibercept arm in that study. The second thing, which I think is very interesting, and we're going to pay close attention to in the next readout, is we had a pre-specified split of looking at patients by disease duration because of this knowledge that senescence burden accumulates over time. And we find that the patients with over 2 years of disease did significantly better than the newly diagnosed patients. So I would say that the emerging view of UBX1325 and AMD would be the patients who have had 2 years of disease or more are most likely to benefit. So going into the Part B readout, we're going to be specifically paying attention to see whether that is also true in Part B of the study, and whether adding UBX to aflibercept might provide benefit above and beyond what they got with aflibercept.
Understand. And speaking of that combination, if it does in fact show the greater benefit in that wet AMD study, how will it affect your thinking about the DME?
Yeah, so this is the first time we're testing the idea of a combination in any indication. So the AMD design, I think, data set will be interesting because they'll tell us whether or not there's a signal or an indication that UBX might add on top of aflibercept. And if that is the case, then it is important both for the AMD study and for future studies and other indications. So now talking about the Aspire study, or what it means for DME patients, the study as designed is just a head-to-head monotherapy study, as I mentioned, right? And in Behold, likewise, it was a monotherapy study design. If there is a signal that indicates that there might be some additive benefit in AMD, of course, it would increase our interest in exploring that in a future study in DME as well, because maybe the patients could have further benefit if you added both compounds. It's not in our current plans, but something we would consider. I think that in AMD, if there is not a clear benefit of combination, I think what it might say about combination DME is still somewhat limited, given that in DME in general, we have seen really strong responses to UBX monotherapy, right? About 6-letter gains. And in AMD, we might not have the same kind of response. So it is possible then in a disease such as DME, where this pathway seems to be particularly important, it could be that there is a benefit of combination, which we might explore down the road.
Okay, got it. So to summarize the clinical program, what are the next upcoming milestones?
Yeah, so the next readout is going to be Part B of the AMD study called Envision, which we're expecting in the second half of the year. Like I said, operationally, the study has continued really well. And so we're looking forward to that data set, and also looking closely to see whether or not disease duration was a big predictor of a treatment response, which might influence our future development in AMD. For now, we're really focused on the DME study and the next DME study, which is Aspire. We are going to share data from Behold at ASRS, which is coming up in just about a week, with some additional analysis included. And in Aspire, of course, it is the head-to-head study. Our expectation is that we anticipate having 16-week data from that head-to-head study before the end of next year, and most likely 24-week data in Q1 of 2025. So those are the upcoming readouts for Aspire.
Got it. Okay, so in the last couple of minutes that we have a couple of general questions. First, if you could comment on the company's cash balance and what do you expect the runway to carry you through?
Yeah, so our last reported cash was about $83 million. And that currently funds our operations and extends our runway. We've guided publicly that has extended our runway through into Q4, so about a year and a half out, which is a good cash position for where we are now. And of course, we'll have our eye towards needs and opportunities as we go forward. So I think that's kind of the cash position right now.
Got it. And finally, the question that I like to end our sessions with, what do you think the investors are missing about Unity's story?
Yeah, I think it's useful to see Unity's data in the context of other studies and other programs that are in development, and what a new molecule in this space might mean, and the value that eventually physicians, patients, and potential partners are likely to see. So the field is very heavily dominated by anti-VEGF dependent treatments, as you know, and as you'll see. And those mechanisms really do not have the opportunity to treat patients who are not responding well to anti-VEGFs. So there is this big population that is not well served by anti-VEGFs, sizable population where a new treatment could have huge benefit. Also, among patients who are responding well to anti-VEGFs and are responding to some extent, going to a new mechanism that can provide significant gain in durability in the ballpark of what we're talking about could be very valuable. Like right now, we're looking at durability aspirations that stand in getting half the patients to 4 to 5 months kind of treatment interval, which is considered a strong achievement. Our data set that gets half of them to 1 year, I think, is really exciting and would be very valuable. I think the other thing that's important to remember is that this field tends to lean in and switch to new treatments very rapidly and much more, much more aggressively than in other fields. And frankly, the story of aflibercept and ranibizumab or Lucentis illustrated that. So, you had a mechanism that was an anti-VEGF dependent mechanism that really does not have any significant letter gains, has some durability benefit. And the projections were, well, patients are doing pretty well on their anti-VEGF, and maybe some of them will switch. But over a very short amount of time, there's a pretty rapid switch to try to get patients onto that little benefit, that additional benefit that patients would get. And even now in practice, and you see this all the time, the reason those drugs are so successful is that patients and physicians are very interested in getting patients to the highest impact drug that they can. A lot of patients start on Avastin, for example, as you know, and then they switch to aflibercept, right? Mm-hmm. Our expectation or modeling would be that if you have a treatment that is providing, as you expect, letter gain compared to aflibercept, potential durability gain, that there is an entire likelihood of a much significant movement to that new treatment than what we and others tend to model. And if you look at Analyst, they'll say, well, some small fraction of whatever the existing treatment is might go to a new mechanism. The reality is that I think if you get to a profile like this, and the history in this field is that physicians will move to this probably at higher rates than people tend to model. So I think it could be very valuable, and that might be something that is not fully penetrated the investor community yet.
Got it. Excellent. Thank you. Thank you very much, Anirvan. It was a pleasure having you at our summit, and appreciate your time.
Thank you so much for having me.